Right then, this is Year 2 Psychology, and this is Biopsychology, and we're looking in this video at functional recovery, which is ways in which the brain is able to recover after it has been damaged. So it's kind of similar to brain plasticity, in terms of we're looking at ways where the brain can change as a result of experience, but we're looking with functional recovery particularly at where there's been a traumatic injury. So if someone had a head injury and part of their brain has been damaged, or if someone has had a stroke where the blood supply is cut off to an area of the brain and it dies, can that person regain the function that they've lost? So for example, if I were to have a stroke on my left cortex, in my motor cortex, the part that controls my right hand, I lose that function completely. I can no longer control my right hand. Are there any ways in which the brain can recover that function of being able to use my right hand? And the answer is yes, because we know that often stroke patients can regain function. So this video is going to look at how well established this is, this process, and how it actually happens. So let's start with the different processes that can occur in order to recover from a brain injury. So the first thing that can happen is something called neural reorganization. So rather than any kind of repair of the damaged brain tissue, this is simply talking about where functions that have been lost are moved to a different part of the cortex. So rather than the brain growing back or anything like that, that function that was happening, for example, my hand was being controlled by the left motor cortex, it would just be controlled by a different part of the cortex. So that's the first thing that can happen, neural reorganization. The next thing that can happen is neural regeneration. And this is where new neurons and new connections are formed. And there are different ways in which this can happen. So I've put down three different ways. The first way is neuronal unmasking. So if you remember when we talked about brain plasticity, we talked about the fact that the strength of synaptic connections can increase and decrease. So where you've got a synapse that's lying dormant, it's maybe got only one vesicle left and one receptor for the sake of argument, that particular synapse can be reactivated and start being used, and then it would increase because it's constantly being stimulated, so it would become stronger and stronger over time. So those dormant synapses are being used again, and that's called neuronal unmasking. The next thing that can happen is called axonal sprouting. And this is where new nerve endings can sprout from undamaged axons. And that's what this diagram on the right-hand side of the page is all about. You can see on neuron A, it's been damaged in the second picture, and so that pathway has been lost. But what's happened, you've got neuron B, which remained undamaged. Axonal sprouting has actually occurred in neuron B, and you can see it's grown new nerve endings, and they've gone over to the first neuron, the first purple neuron, and connected to it, and receptor sites have been created. And a really fancy word for that is synaptogenesis. New synapses have been created. So axonal sprouting, new nerve endings are sprouting from an undamaged axon. Okay, so we've got neuronal unmasking, axonal sprouting. The third way in which neural regeneration could occur, and this is still in the experimental stage really, is through the use of stem cells. Now, if you're not aware, stem cells are actually undifferentiated cells in the body that can grow into anything. So they're kind of like a blank check. And one way in which you might have heard of them is that they're present in embryos, in tiny babies, and they grow into all the different tissues in the body. But they're also present in adults in different tissues. You can find them in the bloodstream, in the brain, in the bone marrow, and so on. And one thing that you can do is actually implant these stem cells into the brain, either to grow into new brain tissue or to help heal the old tissue. Healing is one of the key things that stem cells do in our body all the time, and that could happen in the brain, at the particular site of injury. They could help to heal the site of injury, or equally they can actually, because they've been implanted into the brain and they're an undifferentiated cell, they know that they're in the brain, they can grow into a new neuron, and that is a type of neural regeneration. New neurons are forming. So that's the third way that can be brain injury can be repaired, that functional recovery can occur. Now let's have a look at evaluation. What support do we have for these ideas? And the first experiment that we've got is related to stem cells. Tajiri took rats with traumatic brain injury, and he either injected them with stem cells, or he injected them with a solution like a saline solution that would do nothing. So he had two control groups. The experimental group had stem cells, and the control group had a saline solution. 3 months later, what he found was that the stem cell group had clear development of neuron-like cells in their injured area, which the control group didn't have. So you can see there, that's an example of stem cells having a really good effect. That's obviously quite an experimental thing to be doing. So I'm not sure how far that's gone in humans yet, but certainly it had quite clear results in this particular experiment, which suggests that hopefully in the future something like that may be able to be developed for humans. Right, the next experiment that supports this whole idea of functional recovery is Hubel and Wiesel. They actually sewed one eye of a kitten shut. A bit mean, but there you go. And what you would expect to happen is that the corresponding areas of the brain would be idle rather than processing information, because that information isn't there from the eye. The information carried to the areas of the brain by the optic nerve, you would expect them to be doing nothing. But instead what they did was they simply started processing information from the open eye. So that supports the idea of functions moving around in the brain. It's not like a fixed thing where you have specific areas of the brain can only process that function. The functions can be moved around different areas of the brain if the area where they came from has been damaged. So that's why that supports the idea of functional recovery. Right, more research support. There's a lot of anecdotal evidence and a lot of research that supports the idea. I mean, it's quite well known that stroke patients can recover function after having a stroke, but with a lot of therapy and a lot of time as well. But we've got a few more here, and you'll find these in your booklet expanded in further depth, but I'm just going to touch on them here. And Elbert found that neural reorganization is much greater in adults than in children, and that adults needed extended practice in order to produce changes, but they did happen. And Schneider found that college-educated students were more likely to recover from a brain injury than those who weren't college-educated, and that 27% of them were disability-free after a year. So both of those experiments really support the idea of functional recovery. They support the idea that it takes place across the age range and with lots of different people, that people can completely recover function after suffering a brain injury. However that happens, it is happening. That's well established. And the big thing to discuss here is that actually there's many questions that still remain. Why do some people recover better than others? Why do children recover better than adults? There's still quite a lot really that we don't understand about this process. We obviously can't do experiments on humans. The experiment with the rats and the stem cells, you can't just take humans and try out stem cell injecting them into their brain, because it could go so catastrophically wrong. So for ethical reasons, it's very difficult. Any human research where you've got something like this happening, it's never controlled. So if you take someone with a brain injury and study how they recover, you don't know what their pathology was like before, you don't know of any pre-existing conditions and so on. You haven't observed their brain under normal conditions to see how it reacts. So you've got various difficulties there. And much of the research, as I'm sure you noticed, is animal-based. So Tajiri, Hubel and Wiesel on rats and kittens. So you've got the age-old problem of we can't then generalise to human brains without qualification. So that is the area of functional recovery after trauma. You've got lots of supporting evidence, but also still some questions that remain about the process.